carbon units can also be used to make fat. So you can see this interconversion here. So when you eat a lot of fructose and candy and sugar, that can easily be converted to fat stores. But conversely, when you eat fat, typically you're either going to store that fat or you're burning it as energy, so it gets broken down to carbon dioxide and ultimately excreted as water. All right, so the different processes that we have to learn about in this chapter include glycogenesis, glycogenolysis, gluconeogenesis, lipogenesis, and lipolysis. So glycogenesis is formation of glycogen. Glycogenolysis is the breakdown of that glycogen. Gluconeogenesis is the new formation of glucose. This is formation of fat, and this is breakdown of fat. And fat, in this case, is triglyceride. So remember, triglyceride has the 3-carbon glycerol backbone together with the fatty acids that are attached to it and esterified to it. All right, so review, we talked about the Krebs cycle, glycolysis, and electron transport previously, and we saw that the most efficient production of ATP occurs when tissue has oxygen around, because when it goes anaerobic, then you're relying on mostly glycolysis, because the other two pathways typically are going to slow because not enough oxygen is around to serve as the ultimate electron acceptor in the mitochondria. So you form by far more ATP when tissues have enough oxygen. So this just reviews glycolysis, the Krebs cycle, and electron transport. Remember, glycolysis occurs in the cytoplasm, the Krebs cycle within the mitochondria, and electron transport. Remember, you have on the intermitochondrial membrane the electron transport chain, and oxygen serves as the ultimate electron acceptor, so you form metabolic water, and then via the formation of that hydrogen ion gradient, you're able to form ATP with using ATP synthase. So in the fed state, typically the fed state is going to be defined by the presence of insulin. So insulin tends to favor the formation of glycogen. Amino acids are built into proteins, and those pathways are going to be stimulated so that you're taking all of the nutrients that have come in from the GI tract, and you're building these materials either in the liver, or in the case of fats, you form chylomicrons. The chylomicrons are absorbed in the lymphatic system, and then they deliver the chylomicrons deliver the fat to triglyceride cells in the body, the fat cells in the body, or to muscle tissue, and the chylomicron remnants then go back and the liver takes those up. So this shows the different pathways. So typically, insulin favors the formation of glycogen, so it's going to stimulate this reaction, whereas glucagon is going to be the hormone that's present in the fasted state. So that's going to favor the formation and the breakdown of glycogen into glucose and the breakdown of other foodstuffs into their individual components. So we talked about fat metabolism before. Remember, the liver produces a lipoprotein particle called VLDL, which is very low density lipoprotein. That VLDL in the circulation gets converted to LDL. HDL is also produced by the liver as well as the intestine. You have cholesterol being excreted within the bile, and it's also packaged in the VLDL. And you have triglycerides, which is the main storage form of energy in the body. So typically, the most healthy depot of the triglycerides is going to be found in the subcutaneous fat compartment. So you see there you have chylomicrons that are produced in the intestine. So what do chylomicrons contain? They contain triglyceride, they contain cholesterol esters, some free cholesterol, but they're very large particles, much larger than either VLDL or LDL, and they then enter into the circulation in the venous system. And you can see the different enzymes that are key to breaking down different components of the chylomicrons. You have lipoprotein lipase, which breaks down and converts the triglyceride into fatty acids, which can then enter into the fat cells. So you see in the adipocytes, typically, how do you get fat cells to release energy in the form of fat? You have glucagon being around, which is going to tell the fat to break down, so you get lipolysis occurring, which allows the fatty acids and monoglycerides to then go into the circulation. All right, so the fasted state, as I said, is defined by increases in glucagon, so pathways shift to maintain energy for metabolism. So it's typically going to try to increase the concentration of glucose found in the blood. So insulin tends to decrease the concentration of glucose found in the blood. And the liver is one of the main organs that responds to those hormone signals. So remember, the pancreas, the islet cells of the pancreas, the beta cells produce the insulin. The alpha cells in the islets of Langerhans produce the glucagon. And remember, when you have extreme high-fat diet or high-protein diet, that leads to excess ketone bodies. So the ketone bodies can be used by certain tissues like cardiac muscle as well as others for an energy source. And the brain ultimately needs, certain areas of the brain absolutely need glucose, but some other areas of the brain can function on ketone bodies. But your blood sugar concentration, approximately a teaspoon of glucose in the entire bloodstream, has to be above like 30 or 40, otherwise you start getting what's known as hypoglycemia. So hypoglycemia is when your blood sugar gets below a certain level. That causes your body to go into certain effects. You have shaky feelings, you start getting ravenously hungry, and the body tries to increase the glucose production by organs such as the liver and the kidney. But if that doesn't occur, you're potentially going to pass out. So in certain cases, individuals who have type 1 diabetes and inject themselves with insulin, their blood sugar can go down precipitously if they don't eat a meal after taking that insulin. And if they get hypoglycemic, they can literally pass out, and potentially, if they're driving, they can cause an accident. So as I said, the beta cells produce insulin, the alpha cells produce the glucagon, delta cells in the islets produce somatostatin, which decreases the gastric secretion. The pancreas, we talked about when we went over the GI tract. The pancreas has two different functions, the endocrine pancreas and the exocrine pancreas. So the endocrine pancreas is secreting the hormones that I just mentioned, including, I believe, another called amylin. You also have the exocrine pancreas that secretes all the digestive enzymes together with the bicarbonate that's going to neutralize the stomach acid secretion. So there you see the beta cells are the ones that secrete amylin and insulin. So what does insulin stimulate? Insulin tends to drive down glucose concentration, and it drives down things that can be used for energy, like triglycerides or things like ketone production. So it increases protein, fat synthesis, glycogen synthesis, and you start burning glucose, so glycolysis is stimulated. Whereas in the fasted state, you have glucagon being dominant, and glucagon tends to increase blood sugar. So glycogen is broken down, you form new glucose, or you increase ketone production. And remember, ketones can also be used by certain tissues for an energy source. All right, and this shows, if nothing else demonstrates that insulin is an anabolic hormone, a picture of this one individual who's a diabetic who's been type 1 for a number of years. And he typically injects himself in the stomach. And he did so varying it from one side to another over the period of decades. And when you look at the effect of that, you actually see it looks like he's got increased deposition of fat in the abdominal region. So you can't tell if this guy is literally coming or going. He looks the same both ways, so it's pretty amusing, but not to him, obviously. All right, so insulin action on cells. Insulin stimulates glucose uptake in most cells. It increases glucose use and storage. Remember, what does insulin do? Insulin stimulates the insertion into the cell membrane of the GLUT receptor, so it allows glucose to go through the cell membrane. So in effect, in most cases, when the patient is not diabetic, glucose is going to be a penetrating solute because it can get freely into cells in most individuals. So insulin also increases protein synthesis, and it definitely increases fat synthesis. So there it shows insulin binding to the receptor stimulates a tyrosine kinase, so you get phosphorylation of an insulin-responsive substrate. The GLUT receptor gets inserted into the cell membrane, so second messenger pathways are stimulated by that phosphorylated proteins, which then stimulate other changes in the cell. And I think this is a good place to stop, so I'll pick this up next time.